All right, I'm Jake again. This time to introduce Ryan. Ryan studied plant and fungal biology at UC Berkeley and worked with fungi at Zymergen, am I saying that right? Before joining the Brunkard Lab in 2021. Your title got a haircut.
Yeah.
He's received an NSF graduate research fellowship and an HHMI Gilliam fellowship to support his pioneering investigation of the functional genomics of the quote survivalist Australian outback genus Grevillea. Grevillea.
Yes.
There we go.
Yeah.
Take it away.
Thank you for that introduction. Hi, everyone. Today I'm going to talk to you about the current status of my dissertation research into the genome of a species called Grevillea lanigera that's native to Australia. Before we get into that, I'll explain why I care about this random plant that no one has heard of. The three main macronutrients in agricultural fertilizers are nitrogen, potassium, and phosphorus. Phosphorus is a non-renewable resource. We mine it from the earth, and once we mine all of our phosphorus, there's not really a plan for how we'll address when we run out of phosphorus. It's estimated that we will run out of phosphorus, or at least we will reach peak phosphorus production sometime before the end of this century. And so there's not really a plan for how we'll address this issue. And then, as people who live in Wisconsin, we get a lot of algal blooms in the summers, and that's largely in part to agricultural runoff that contains a lot of phosphorus. So the overall goal of my project is to identify ways that we can improve phosphorus use efficiency in plants that could maybe decrease our reliance on unsustainable fertilizers. Also, yeah, so my presentation got a haircut, and so I converted this to PowerPoint, and so some things might be a little off. I think aside from the title, it should be okay, but yeah. All right. So Australia is home to some of the most phosphorus poor soils in the world. Most of their land is not arable, and it's been largely undisturbed for the last 90 million years, which has led to a lot of nutrients being leached from the soils. This map here shows the global distribution of phosphorus around the world. You can see in the Midwest, our soils are quite fertile. Across the southern hemisphere, we see a lot of low levels of phosphorus, but Australia is cool, so we study Australia. And so advances in sequencing technologies allow us to start understanding, how plants from non-model systems have really adapted to grow well in this environment. And so I'll introduce my plant family, which is called the Proteaceae. These are their flowers. I think they're very charismatic. And so the Proteaceae are like a very large family. They have their greatest diversity in Australia. In particular, Grevillea, which is my species, is the largest genus within Proteaceae. But I just thought I'd include their flowers because, it makes the presentation a little more interesting. We know a lot about how these plants have adapted to growing in low phosphorus conditions, but we don't know a lot about what's happening at the genomic, transcriptomic, multi-omics level. And among those are some physiological and morphological traits that have been characterized. And so at the top here, we have some photos of roots. So what Proteaceae do is they form these highly clustered root systems called proteoid roots. And what they do through them is they, first they increase the surface area of their roots, and they also secrete organic acids that allow them to fix and mobilize phosphorus that's otherwise unavailable in their environment. And then I also want to bring your attention to this pie chart that was made by a postdoc in our lab. And so what this is describing is the allocation of phosphorus in Arabidopsis. And so what we see is that, one thing, plants and, all cells do is they have like a phospholipid bilayer And one way that proteaceae can decrease their phosphorus use or phosphorus needs is by substituting their phospholipid bilayers with galacto and sulfolipids. Another thing they do is they delay when they develop their photosynthetic machinery. And most importantly, or at least in the context of our lab, they actually, at certain points in their development, they decrease ribosome abundance by up to 40-fold. And so this chart on the purple rRNA would be ribosomes. So, we know that these plants are have low levels of ribosomes. And so they're really doing the most when it comes to decreasing the amount of phosphorus that they can require from their environment. Oh, I bolded it because it's important. And so as Aaron explained earlier, our lab studies TOR. I'm not going to repeat too much, but except for the part that we know TOR is essential for promoting ribosome biogenesis. And typically in model systems, when you perturb ribosome biogenesis, you get aberrant growth phenotypes like dwarfism or chlorosis or like weird leaf morphology. And so I'm interested in also understanding how these plants are able to grow with low levels of ribosomes and what TOR is doing in these plants. That's why are there few ribosomes? What is TOR doing? And what kind of proteins are they making in order to allow them to grow without weird phenotypes? Okay. And so largely what I've been working on up to now, I'll talk about the project I'm starting this next month. But currently, I'm finishing up defining what the Grevillea genome looks like. And once I have this, I'll be able to proceed into my next studies. But right now, I am assembling and annotating the first high-quality reference genome for Grevillea. It's the largest species in genus in Proteaceae, but there are no reference genomes. There are other Proteaceae reference genomes, but no one's done Grevillea. I don't know why. My investigation is mainly interested in understanding how a phosphorus-poor environment has influenced genome structure. And so briefly, I will go over my genome assembly pipeline that I stitched together. So what I did was I used long reads from 2 platforms supported by the sequencing facility down here. So we use PacBio and Nanopore. PacBio reads are very high quality, they're long, and you can get some pretty high quality genomes. Nanopore reads are nice. They're a little longer than PacBio, however, they're not as high quality. And so what I can do here is assemble 2 genomes using both platforms and then improve the Nanopore assembly with my higher quality PacBio reads. Okay, I don't know how to use this thing. Okay. Polish the Nanopore assembly using my PacBio reads, and then there's a lovely little software called QuickVerge that allows me to take the best parts of both assemblies and make a nice consensus assembly. From there, I can remove some spurious sequences that might come from sequence over assembly. I'm not going to get into the nitty-gritty of that. But once that's done, I can use, or what I'm currently working on is incorporating some Hi-C data or OmniC, which is like Hi-C, except it's using a different enzyme for DNA digestion. And so what OmniC does is it just captures how DNA is interacting in 3D space. With that information, you can sew your genome or your contigs together into a chromosome level assembly. And so with a collaboration at the University of Delaware, I have some OmniC data that I'm working on, working into my genome. I'm waiting for some more data to come back from them, but that is currently where this is. And then once that's done, I'll be able to generate a chromosome level assembly. In the lab, we have 2 accessions of Grevillea lanigera. So these are just essentially just 2 different genetic backgrounds. I have one on the left, I always have to do that, called Mount Tamboritha, and then one on the right called Coastal Gem. I picked 2 strains because I wasn't sure which one would be the easiest to grow. And we actually killed a lot of plants in figuring out which Grevillea species to grow, and this is just the one that's stuck. So that's why I have 2 genomes. But that leaves a lot of room for understanding chromosome inversions or deletions. what families of genes are present in one accession, not the other. And we can also figure out when these two accessions diverged. But that's also not the point of my research, but a nice little thing I can tack on. So these are the just some assembly statistics for my draft genomes. They're pretty highly contiguous. So in the 60s, they did like they estimated the size of Grevillea genomes. They just said Grevillea. They didn't even say like which genus or which species it was. There are hundreds, but conveniently, it was about 830 megabases is the genome size was estimated to be. My genomes fall between 860 and 830 megabases, which is a nice like sanity check, like that I'm doing something right. And then also when I ran Busco, which is a software that essentially looks at your genomes and scans it for genes that should be predicted in like a core, like a core set of genes that you should expect in a certain like type of organism. You could do plants or E. coli, what have you. But my scores on a scale of 100 are 97 and 96 respectively, which is honestly like that's about as good as it can be. Like compared to some of the other published protease reference genomes, it's a bit higher than them. And it essentially any other genomics paper like this is pretty like good. So I know I'm capturing most of what's happening in genomic space with respect to protein coding genes. And so that is where these assemblies are right now. Currently, I'm working on assembling this, or as I mentioned, I'm working on scaffolding my assembly. So this is a Hi-C interaction map. And so the X and Y axes are showing the same genome. It's essentially just a matrix where, one position is on, or the genome is on one side, same side. And this is showing how the DNA is interacting in 3D space. And so what you should expect is that on a chromosome, or like a piece of DNA on one chromosome should not be interacting much in 3D space with a piece of DNA on another chromosome. And so you can see that there are still some like interactions just due to like, how DNA exists in the nucleus. But for the most part, DNA interacts quite like discretely within its own chromosomes. And with these data, what we can do is scaffold our DNA or our genome into chromosomes, and we get these lovely 10 little boxes. And so for multiple Grevillea species, they didn't estimate the genome size, but people have assayed ploidy, and they estimate for, I think, about 30 Grevillea species, there are about 10 chromosomes, or at least for like haploid karyotypes. And so like this is also at least another sign that things are going well. Oops. And so once this is done and I have my genome at a chromosome level, I'll be able to generate some lovely Circos plots. If we do journal clubs in our lab, I like to do genome papers. I love these figures. But essentially what this is just showing is like different pieces of information, different layers showing like tracks of like repetitive elements or where genes might be in your chromosomes. The circle is just following the space of the genome. And then the little bands in the center are showing like syntenic duplications. Yeah, that's actually just it. And so the next thing I'll talk about is my genome annotation pipeline. And so although my genome is not at the chromosome level, I'm still able to annotate genes in my draft genomes. And so what I did was I took long-read RNA-seq data, and then I also grabbed a bunch of protein FASTA sequences from other closely related species and model systems. And what I can do there from there is run them through different gene predictors. And so I ran them through 3 gene predictors, one focusing on just using the RNA-seq data to annotate, one called Breaker3, which uses both and some machine learning magic, and then another one called GMOMA. I don't like how they name these, but it is what it is. That uses just protein data to predict genes in your genome. And then from there, you can use a software called Evidence Modeler to generate a consensus and figure out, what genes are the most real, considering the evidence you have based on your 3 assemblers and your 2 different data types. So that's the structural annotation portion of my research. And then once that was done, I ran them through a software called InterProScan, which does functional annotation. So now that I have all my genes, I can assign them functions to figure out, what's related to phosphorus use or what's related to ribosomal protein genes. And so this is what I have so far. So I've annotated 31,532 genes, which is pretty close to what we should expect for other Proteaceae species. The genome is 64% repetitive elements. Plant genomes are largely repetitive, about 65% of them, or like this is usually what the repetitive content is, and then 21% is long-term repeats. And now that I've done this, I've also started to dabble in the comparative genomics part of my project, where I'm interested in understanding how phosphorus has, how growing in Australia has affected the genomic landscape of Grevillea. And so the first thing I did was grab 23 species representing broad or, multiple parts of the plant kingdom, from early diverging eudicots where Proteaceae are, to model systems, which largely consists of the core eudicots, and then monocots like maize and grasses, and then early diverging plants or the earliest evolving plants like mosses. And so what I have here is a heat map showing the copy number variance, or a copy number analysis of a couple hundred genes involved in phosphorus use in plants. And so red here would indicate that, or I'll first also mention that the x-axis is just lists of gene names. It doesn't include all of them just because it wasn't really practical to fit them all. But essentially, if you look at it at a column perspective, a red gene would indicate that this gene has higher copy number in one species, and a blue gene would indicate that it has lower copy number. And so ideally, I wanted to see Proteaceae have this crazy red cluster that isn't present in other species. And that's not what I've seen so far, which is fine. But when I applied hierarchical clustering to my species, I found that we can at least group Proteaceae together, which would at least support that their phosphorus use machinery is shared ancestrally, which would make sense. And although there's not really anything really jumping out to me right now, at least with my first pass, it is nice to see that they are grouping together. And that would maybe suggest that their regulation of phosphorus use is more related to something happening at the transcriptional or translational level. And so my next part of my project will get to that. And then somewhat unrelated, but a nice sanity check, was that we know based on other Proteaceae genomes that they've lost the ability to form arbuscular mycorrhizal symbioses, and based on the literature. And then when I looked at what was absent in my genome, I also found that my Grevilleas did not have these same genes required for symbioses, which was a nice sanity check, and I just thought it was interesting. And also, these genes are present across all types of plant species. So, it's very, very unique to Proteaceae. And so when the orthogroup analysis didn't really throw out anything spectacular, I used another tool called Cafe5, which calculates gene family expansions and contractions. And so when I used the same panel of species to figure out which gene families have expanded in Grevillea, I did find that the genes that have expanded are functionally enriched for flavonoid biosynthesis, cell wall modification, and oxalate metabolism, among other things. So flavonoid biosynthesis is important for stress responses to abiotic things. Oxalate metabolism would be organic acid secretion. And we know that these plants form proteoid roots that are essential for secreting organic acid. And so it's nice to see that, although I couldn't immediately identify this in my orthogroup analysis, statistically, we are seeing that there are gene family expansions that are that support my hypotheses that growing in a low phosphorus environment has caused these plants to, expand gene families or that confer higher PUE. And then let's see, how am I doing on time? Oh, I'm fine. Okay. So the next part I'd like to touch on, NIH says that we have to talk about responsible research conduct, and so I will talk about that. I have to do a lot of data management and use a lot of softwares to do this project. And so to make sure my research is reproducible, all of my sequencing data will be available on SRA, and my genome will be available on NCBI following publication. All of my software versions are recorded. I find that a lot of times in research papers, people forget to cite the version of the software that they use. And I've spent many days trying to figure out which version of software is to use. So that's something I do. And then also I annotate my Python and various scripts very well, and they're all made available on GitHub. And yes. So my comparative genomics analysis has just begun this summer. I have a lot more that I plan to do. I eventually plan to characterize whole genome duplications in Grevillea and other Proteaceae species to see, how that has been happening. And I also want to measure amino acid substitution rates and see how genes are evolving. And then because, our lab studies TOR, and TOR promotes ribosome biogenesis, I'm also interested in understanding how ribosomal protein genes have evolved in this species. And so we care about ribosomal protein genes because in mammals, we each have we each have one copy of a ribosomal protein gene. And there are 83. In plants, this is highly variable. It can be upwards of it can be as high as or as low as 200 or as high as 500 based on what I've seen so far. And so I'm interested in doing some analyses to see if Proteaceae, given that we know that they have lower levels of phosphorus, if they also have lower copy number of ribosomal protein genes, which could maybe explain why they have fewer ribosomes, and also measuring the amount of ribosomal DNA in their genomes. So once this is done, next month I plan on starting my hydroponic stress tests. And so these are not my Grevilleas, but they are some Grevilleas from a paper in 2006 where some researchers grew their Grevilleas, different species, but they grew them under different levels of phosphorus to see if they could induce proteoid root formation. And so this was great because, using hydroponics is a great way to really tightly control the level of nutrients you're providing your plants through liquid media. And so what I'm going to do here is grow my plants hydroponically and collect RNA and polysomes at different concentrations for different tissues. So I actually had to grow these plants by my bedside because I was paranoid that they needed a lot of care, and they did. But eventually I was able to propagate them successfully. And so once these cuttings are old enough, I can shake off the dirt and then transfer them to their little hydroponic boxes where I can carry out my plan. But what I'll do is I'll collect roots and leaves. For roots, I'll collect proteoid and non-proteoid roots because it'll be really interesting to understand what are the genes responsible for root cluster formation. And then I'll carry out differential expression analysis using or to figure out what's up and down in different conditions. And I'll also incorporate some network analysis to try to identify some central hubs that might be involved in the regulation of the phosphorus stress response. And then we'll also measure TOR activity in these different conditions to see, is the level of phosphorus affecting how these plants are growing, and if higher levels of phosphorus are activating TOR or deactivating TOR, or if it's not changing at all, because it's really no one knows. And so, yeah. And so we know a lot about RNA-seq, but I'll briefly touch on polysome profiling, which is translatomics. This is a figure, at least the nice part on the middle and left side is, I made the other part, but a postdoc in my lab made a really nice figure explaining polysome profiling. And so what you do is or first I'll start with a polysome is an mRNA transcript that has two or more ribosomes bound to it. Transcripts can be really highly transcribed, but not necessarily translated. And so we're really interested in understanding which transcripts are the most highly translated in addition to the most highly transcribed. And so what you do is you extract them and then you separate them down a sucrose gradient, and you can run them through a fractionator that can quantify like the amount of ribosomes or RNA you have in each part of your profile. From there, you can split your little graph into fractions and send them for sequencing and determine which transcripts are more highly transcribed. So that's what we will do eventually. But what we've also shown so far is that translation occurs at very low levels in my plants. And so on the left, we have Arabidopsis, and it's very happy. It's translating really high levels. There's nice discrete peaks showing ribosomal subunits and monosomes. And then as we move towards the right, we can see like, peaks corresponding to like this peak would be two ribosomes, three ribosomes, and so on. In Grevillea, we see some peaks, but largely like, we see these tiny peaks showing that, there's not a lot being translated. So I'm very interested in understanding, what proteins is Grevillea translating at low levels that allow it to grow really well, which opens the door for a lot of questions. Maybe, these proteins have longer half-lives than other proteins. But that's what the next part of my experiment will get to. And then I'll also start doing network analysis. This is unrelated to this is an unrelated thing or network module. But what we're showing here is that these genes that are in the red are really downregulated under one condition. All together, like they're all co-expressed in the same way. They're all down under TOR inhibition. And then we see the blue triangles are transcription factors. And when we see that they're blue, they're going up. So it's interesting that these transcription factors are up and the ribosomal protein genes are going down. So this is just like be going one step beyond differential expression analysis and trying to get maybe more of like a clear picture of how like these plants are coordinating growth and development under different phosphorus conditions. And so in summary, I'm working on 2 draft genomes right now, very close to finishing them up. And then after that, I'll start assaying gene expression dynamics. And yeah, that's it actually. And so I'd like to acknowledge my lab and our collaborators and my committee and my funding sources. Yeah, thanks. Oh, yeah, this talk, I can't see who is raising hands. Oh, sorry.
You might not know this, like you kind of mentioned it with specifically with respect to SORT, but do you know how plastic the response to like either phosphorus stress or abundance is on like what kind of phenotypes they have?
Yeah, so Myron is up for the Zoom. Myron is asking how plastic the phosphorus response is to the Grevillea phenotypes. And so we know that like higher levels of phosphorus actually induce a toxicity response. This species from this paper actually has a little bit more of a tolerance to higher levels of phosphorus, but by and large, Proteaceae generally, like when they're given higher levels of phosphorus, they start to show like signs of toxicity. Yeah.
Do you know if there's any record with their tolerance to like other nutrients like nitrogen or?
Someone asked me that in my committee meeting like last week. I'm sure there is. I've definitely read reviews that talk about this, but off the top of my head, like I don't know. Yeah. Yeah.
There's an interesting pattern in your Omni-C data. You had this really nice cluster showing the different chromosomes. But
Yes.
they almost look like cruciform patterns. Has anyone looked at how the chromosomes are organized inside a nucleus? Do they form rosettes? And that's why you're getting telomeres talking to telomeres type of thing.
Unfortunately, I don't know. So one issue here or with these data is that the sequencing coverage is about half of what I needed to be. So Delaware sent me the data, and then I did like the PCR, the deduplication, and like I have requested like another round of sequencing just because the sequencing coverage is just insufficient to resolve some of this stuff happening. Are you referring to like the bottom right corner?
In each box, there's a diagonal going from the upper left to the bottom right, but it also looks like there's a diagonal kind of
Oh,
moving in the other direction. And so that would be suggestive of like pattern organization of the chromosomes inside the nucleus.
oh yeah. So I don't know, but yeah, my best guess so far is that it's like some sort of centromeric thing, but I haven't been able to really get into like what's happening yet. Francisco, or oh.
So this so like left level protein that could change, right?
Yeah.
Regulation. I know phosphorylation is very important.
Yeah.
I wonder, do you think that the phosphorylation of the regulatory signal might be different?
I am sure it is. I think like I know Jake has suggested doing proteomics or phosphoproteomics, but it's more just like, will we have the money to do it when the time comes? But yeah, no, I'm pretty sure like there probably is some differential phosphorylation happening. But oh yeah, I mean if there's less phosphorus, I would imagine that they probably are phosphorylating things less. But yeah. Oh, Myron.
Slightly tangential, but I'm just curious if you know, like how widespread is low phosphorus tolerance in, like how far away from the species you're looking to have to get before you start losing phosphorus tolerance? Like how widespread is that?
Yeah, so how widespread is it? The best I can answer that is like Proteaceae are like pretty like they have a large distribution throughout the southern hemisphere. There's one species, this circular one at the top. It's actually native to South Africa. It's actually quite tolerant of higher levels of phosphorus, but it's not really no one's really like looking at this stuff right now, so I can't really like answer that. But that's like as much as I can say. Yeah.
Going to this graph, do you think really the problem is that we are going to run out of phosphorus? Because all the places that has like low phosphorus have, besides deserts, they have high fertility. They got like basically Central South America, lots of parts in Africa, lots of parts in the Asia, all the places that have like low phosphorus, they are getting fertile. And the problem are places that have like high phosphorus. Maybe
Yeah.
they have monocultures, a lot of like.
Right. But the only like yeah, they do have high levels of phosphorus, but even here where we have some of the higher levels of phosphorus in the world, we still have to supply like a lot of fertilizers to our crops. And then most of like the time, like plants only take about 15% of the phosphorus that's supplied to them. Most of it just goes into runoff. So although like they do have higher levels of phosphorus, they're not actually able to use it very effectively. And so, the only reason we're producing more food is because of like the Green Revolution, and like the Green Revolution was like not a sustainable thing. So like, yes, there are places that have more phosphorus, but also in these places where subsistence agriculture is happening, the levels of food production aren't really scalable to like meet the needs of like other parts of the world.